situated with where Bering Sea is. I'm sure all pretty familiar. But Bering Sea is a region over by Alaska, has really cool geography where you have this, this pointer is working, this really shallow shelf, average depth of around like 80 or so meters, and then a really deep basin nearby. So that brings up a lot of just upwell nutrients in this basin, which is really productive, and that contributes to a really high invertebrate diversity in the region. Taxonomists love to disagree with each other, so depending on your source, anywhere between 150, 250 species of gastropods in the area, but the bottom line is there's a lot of diversity. These gastropods, like ecologically, are really good scavengers, cycling nutrients that fall into the bottom. They also act as a food source for some pretty important species. We got on the top here the Pacific cod, pretty economically important species, and on the bottom here we have the sunflower star, which is a really important keystone species limiting urchins in a lot of areas. They also like to eat snails sometimes if they don't have these urchins. So these gastropods are contributing ecologically significantly. Also in the Bering Sea we have some really well-studied fisheries. A good example of this is the pollock fishery, which is, I believe, the most important economically important fishery in the U.S. But these gastropods don't have a fishery since people don't like to eat them. So as a result, they are a lot less studied. A caveat to that is in recent years, NOAA has done some really good groundfish invertebrate survey trawls, which are these yearly assessments where they just take a lot of trawl samples and get an idea of the entire community of Bering Sea. However, this is just in recent years. This data goes back around 10 to 15 years, so we don't have a good idea of like a historical baseline to compare that against, which is where my project is trying to come in, which is trying to get a historical baseline, main objective being to characterize historic Bering Sea gastropod communities. So doing that using geographic data to identify trends in species abundance, richness, and community composition. The data from this is from these old NOAA trawls from the 70s and 80s, which were taken and then essentially just sat in a warehouse for 50 years and were completely unused. Then a few years ago, they were donated to the Burton Malacology collection and then were able to be incorporated into the database and analyzed. So this map up here just shows where they're all taken. This map of Bering Sea and Gulf of Alaska. Each dot indicates an individual trawl. So along with the trawl locations, we have the vessel ID and trawl number recorded, which we can correlate or correspond to ship logs to get pretty precise GPS coordinates of latitude, longitude, as well as depth. We also know the year collected, and then went through all of them and identified them down to the lowest taxonomic ID, usually species level. And so in here we just have some examples of these shells, very dry, fairly old, very old for snails. My next step was then to see do these samples give us a good idea of what we are actually seeing in the Bering Sea? Did that create a rarefaction curve? For those of you that are not familiar, x-axis here we have sample size, y-axis we have species richness. It's essentially saying if we took X random samples from the data set, what's our expected species richness? And as we can see, it's leveling off, which indicates that we have pretty good sampling efficiency reached, even though we're not seeing every single species in the Bering Sea, which I'll talk about later on. There we go. From there, I wanted to see what geographic factors were predictors for abundance, so shell count, how many snails are we capturing. To do that, I created a generalized linear model, and then went through stepwise model reduction, where you essentially test the model removing different predictors each time to eliminate any unnecessary factors. So that found that latitude is the most significant predictor of abundance. Here, the blue line represents the model trend, and the individual dots represent the actual data. So as you can see here, we do see a trend, but there's also a lot of dispersion with the data. So there's other things that are going on here. From there, visualizing the data, seeing trends of species richness across the sample area. This is a similar map, but instead of individual trawl locations, it's a heat map showing species richness. So darker red represents more diverse, more species-rich areas. As you can see, we have sort of three main regions here. We have a group up in like the North Bering Sea around the Bering Strait and the northern Sound, one that's here on the shelf, Bering Sea shelf, and then another group in the Gulf of Alaska. So looking at comparing those specific regions, ran an analysis of similarity test, or ANISIM. These trees here can be read similar to phylogenetic trees, showing with branch length representing like dissimilarity between groups. So we can see that the Gulf of Alaska, the North Bering Sea, and the Bering Sea shelf fit into nice little discrete groups, whether you're looking at abundance, like shell count data, or just species presence, whether or not you see species at trawl sites or not. Then running a SIMPR test, so a similarity percentage, just seeing which individual species significantly contribute to the difference between these regions, found that there was, for both abundance and species presence, there was no significant difference between the Bering Sea shelf and the North Bering Sea, so no species were contributing to that. There's a small difference, around 7 species of the 89 total in the collection, contributing to the difference between the Bering Sea shelf and the Gulf of Alaska, and a pretty large difference, around 25 species, contributing significantly to the difference between the Gulf of Alaska and North Bering Sea. From there, I also wanted to see comparisons with depth. Depth is very commonly used as a predictor for like species richness. We see a lot of impacts of that generally. So defining depth groups by 50 meters, like I mentioned, the Bering Sea is very shallow, so we want to see good gradation in the upper region. So similar idea, making these trees with abundance and species presence data. One thing to note here is that the regional comparison, we saw identical trees with both types of data. Depth comparison is a slight difference. We see that the 50 to 100 meter group shifts from shallow to intermediate depending on abundance or species presence, but the overall results and similarity between groups was similar. Running a Permanova analysis on the depth groups shows they're statistically significant, but don't explain much of the variation, only around 6%. So again, there's a lot of factors at play here. Then the difference between groups here, no significant difference between the shallow and intermediate depths. So the shallow being 0 to 100 meters here, and then just 0 to 50 meters here, and intermediate being 100 to 150 meters here, and 50 to 150 meters here. So we don't see a lot of difference in the top, about 150 meters. We saw a really, really large difference between the shallow and deep depth groups. About half of the species, so like 50-ish of the 89 total species, were significantly contributing to that difference, and then a pretty large difference between the intermediate and deep depths, around 25. Going on, I wanted to look at a specific group. The genus Neptunia was by far the most abundant in this region. Of the 2,200 total shells, approximately 800 of them were Neptunia, and 9 of the 89 total species of Neptunia. So they're very abundant, very widespread, almost ubiquitous across sampling sites. So running models on specifically Neptunia, similar to stepwise reduction like I mentioned earlier. On the top we have a GLM model showing just that latitude is the most significant predictor. So we have latitude on the x-axis here, count on the y-axis. Again, the dots represent the actual data, and the blue line represents the model's prediction. It's a little bit less dispersed than we saw in the other one, and generally sort of similar trend. We're seeing more Neptunia at higher latitudes. On the bottom here with presence data, so y-axis is the probability of finding the genus Neptunia. For that, I found that sort of unexpectedly, longitude was the most significant predictor. So we have longitude on the x-axis here, so more negative longitude here, so going out to sea. Yeah, and we have the dots here representing the actual data, so zeros or ones, sense presence or absence. And we see generally a pretty decent trend of increasing probability of presence of the genus Neptunia as we go to a more positive longitude, so like more towards the Gulf of Alaska. But again, some pretty significant variation in that. I also want to talk about some limitations of the study. Since this is data that I did not myself collect, this is from the 70s and 80s, so there is a pretty big gap between collection and analysis. We also, I'll go back to this in I think the next slide. There's uneven sampling effort. I adjusted for this by accounting for the duration of trawling, but we don't have a lot of samples from like the center area of the Bering Sea, so there is potential bias added there. Another important note is that the trawls used to collect all this data, the mesh size was 5 centimeters, and a lot of snails are smaller than 5 centimeters. So that's one of the reasons that, going back to the refraction curve, we saw that we were getting a pretty representative sample, even though we're not seeing full diversity. We're just not seeing those snails that max out below 5 centimeters. So some reasoning for why we saw some trends here. The big difference between the Gulf of Alaska and North Bering Sea, likely due to the large geographic difference. It's very far, and there's Aleutian Peninsula blocking them. We also see generally a much larger impact of sea ice on the communities in the North Bering Sea than compared to the Gulf of Alaska. And then additionally, the high richness along the Bering Sea shelf is probably just reflective of the upwelling and high nutrient content of that region. So just to kind of reiterate some main points here, the communities vary a lot with depth and latitude, but there's also potentially other factors that are causing changes here. Neptunia genus specifically is interestingly impacted by longitude, so more likely to see Neptunia as we go in here in the Gulf of Alaska than along the outer bank. So future studies, future directions to take this would be comparing it to modern data. There's the modern NOAA trawls that are really good data, but they don't account for like a baseline, so it'd be interesting to compare that to modern day data. I also want to give a special thanks to Lou Trumpene and Melissa Frey for consultation advice and just help with the project. And with that, I'll open up to questions.
Great job.
Thank you.
Do you know anything about the diet of Neptunia? Do you have any speculation about what it is longitudinally that might be different?
The diet, from what I've seen, is they're sort of like all over the place. Just they act as scavengers. They also eat other types of snails. I don't have a great answer for why that would impact them longitudinally. It could just be different prey species or prey availability. Yeah. Oh, question up there.
It looked as though there was sort of spikes in the pattern, and you can fit a line through that. But
Yeah.
I'm wondering whether those were reflecting shelf break or certain features.
Yeah, that's a good question. I don't have a great answer for that right now. I guess more work would need to be done looking at specific things since the data I had was mainly just based on like geographic data. You could definitely try and correlate that to breaks. By shelf breaks, do you mean into the deep sea here?
The latitude and longitude was kind of,
Yeah.
it wasn't as smooth. There was a couple of ups and
Yeah.
downs. I was wondering if you might pinpoint that to some area that for some reason had anomalously
Yeah.
different conditions that might have more of them.
One potential explanation for that is that we see like very little sampling in this region, and that could be the reason.
Sorry, I can't see
Oh,
it.
sorry. Right here we have
Oh, okay.
essentially no samples, so it could be that we would normally expect to see like a very high diversity across this entire area, and we just don't have sample there, so it looks like really high here and really high here. But yeah, there's a number of other things like, I don't know, like sea ice cover that could also potentially explain it.
I'm just wondering, so the counts and whatever you're using,
Yeah.
are those derived entirely from the dry collection?
Mm-hmm.
So they weren't keeping records of how many they were catching as they were getting them or anything like that?
No, we just got the shells that they gave us, so that is another part of the study. It was offset by the sampling duration, so we know how long it took them to collect the number of shells. So we have, like with each set of shells, the like trawl number, and then we know how long that trawl was down there for.
Right.
But we don't have like exact like on-site population counts.
I'm just wondering about how consistent they were about actually collecting them.
Yeah, that's
each trawl.
another potential issue is that it has been about 50 years that those have been collected, so a lot of the people that initially collected them retired, so
Yeah.
it's hard to ask them about that. Yeah.
Yeah, I know that, well, Catherine probably knows better. How many years ago did they add the snail bag?
It was early 2000s,
Yeah,
yeah.
which that was gonna influence too what shows up if they were keeping track of what's the snails, and not just the snailfish.
Right, right. And then you never know for sure, did they retain every snail that they
Mhm.
caught?
Right.
And,
Yeah.
what sort of selection bias is happening? How long did it take you to identify all this?
It took a few months. They were generally put into like lots where each lot represented like all of the things that the people collecting it thought were one species from one trawl. So there's around 800 lots to start out with, but a lot of those lots were also not correctly grouped. So it took, I want to say, like all of autumn quarter and then part of winter quarter to go through all of that.
It's a lot.
Yeah, lots of snails.
All right, next up we have Jason Ray, who's also been working with Tom Quinn, and he's talking about Chinook controllers.
Hi everyone, as Greg said, I'm Jason. I'm talking about temperature